Module G, Power Calculations. This module is quite similar at a conceptual level to Module G from Part 1 of this online training course. Both modules cover the topic of simulation-based approaches to power calculations in clinical trials with multiple objectives, and there will be inevitably some overlap. Just like in Part 1, I will talk a bit about analytical approaches to power calculations that are used in a traditional setting with simple objectives. But when we are facing the problem of power calculations in trials with complex objectives, we have to resort to simulation-based methods. Simulation-based approaches can be applied in a disciplined, structured way using the general clinical scenario evaluation framework, and I will give a quick overview of this general framework. The last topic is the Mediana package that we used in modules C and D to implement powerful gatekeeping procedures in problems with parallel and general gatekeepers. But the main goal of this package is to actually support simulation-based power and sample size calculations in a very broad setting, which includes power calculations in phase 3 clinical trials with hierarchically arranged clinical objectives, and we're going to use the Mediana package to run power calculations in example 3 based on a schizophrenia trial. Section G1, Power calculations in clinical trials with multiple objectives. Let's begin with a general discussion of the role of analytical and simulation-based approaches to power in clinical trials. We know that standard sample size calculations are based on analytical solutions that rely on closed form expressions. In traditional sample size calculations, we typically use formulas to compute the number of patients or number of events in trials with a time to event outcome. The reality is that analytical solutions are available only when we make or we are forced to make simplifying assumptions. Formula-based approaches are applicable only when basic assumptions are made, such as an assumption of a two-arm design or a single normally distributed endpoint. In most modern phase 3 trials, these basic assumptions are not satisfied. Instead, we have to deal with complex trial designs and advanced analysis methods. If we try to force simplifying assumptions, we will run into a problem, and we begin to ask ourselves the following question: are we trying to cut corners and look for shortcuts in multi-million-dollar clinical trials? A viable alternative to an analytical approach is, of course, a simulation-based approach to power and sample size calculations. This approach is much more reliable in trials with complex designs and complex analysis strategies, and we also need to keep in mind that in many complex phase 3 trials, analytical approaches and closed form expressions are simply not available. To support our focus on a simulation-based framework, the FDA's guidance on multiple endpoints recognizes the importance of computer simulations in clinical trials with complex objectives. It is stated in Section 3 of the guidance that determination of an appropriate study sample size to ensure that the study is appropriately powered can be difficult in these cases, and often we will be dependent upon computer simulations rather than an analytic formula, which can be used for simpler situations. This is exactly what we're going to explore in this module. Introduction to Clinical Scenario Evaluation. In this section, I'm going to go over the main ideas behind the Clinical Scenario Evaluation framework, and due to the overlap with Part 1, I will be making multiple references to Module G included in Part 1. The Clinical Scenario Evaluation framework is built upon a set of concepts developed in several recent publications. The term Clinical Scenario Evaluation was introduced in the paper by Benda and other publications, including the paper by Tim Friede and colleagues. The clinical scenario evaluation approach is best described by the following three words that are highlighted on the slide: quantitative, comprehensive, and disciplined. Clinical scenario evaluation enables clinical trial sponsors to perform quantitative, comprehensive, and disciplined assessment of applicable designs and analyses in clinical trials with the ultimate goal of enabling better decision making. As I said, clinical scenario evaluation is a structured approach and it supports workflows that mimic the process of collecting data, analyzing data, and evaluating results in clinical trials. A good analogy to make here is that clinical scenario evaluation is similar to the periodic table of elements that, as we know, plays a central role in chemistry. And the analogy here is that clinical scenario evaluation provides a structured approach to studying trial designs and analysis methods used in clinical trials. This approach encourages clinical trial sponsors to break down a complex problem of assessing all components of a clinical trial to the key elements or steps, such as specification of parameters of the data generation process, specification of the analysis methods, and evaluation criteria that are closely aligned with the clinical objectives of a particular trial. This leads us to the definitions given on the slide. Data, analysis, and evaluation models are the key components of the clinical scenario evaluation framework, and as you can see on this slide, data models describe the data generation mechanism in a clinical trial. Analysis models define statistical tests, descriptive statistics, and potentially other analysis tools, for example, multiplicity adjustments, which is of course the main topic of this online course, computed from the trial data. And the final component is an evaluation model, which specifies measures for evaluating performance of the selected analysis strategies. Later in this module, when we begin discussing power calculations in example 3, I will walk you through the process of setting up these three models, and then we will use the Mediana package to run power calculations based on these models. Every time we talk about applications of clinical scenario evaluation, I like to point out that one of the most important applications is clinical trial optimization. Within the clinical trial optimization framework, we utilize clinical scenario evaluation to help us transition from traditionally used approaches to optimal approaches to selecting trial designs and analysis strategies in our clinical trials. And the overall goal of clinical trial optimization is to inform decision making at different stages of a drug development program to help maximize the overall probability of success. General optimization approaches have been applied to a broad class of problems. For example, problems related to an optimal selection of multiple testing procedures in clinical trials with several objectives. Also, optimal selection of parameters of a multiple testing procedure, such as selection of hypothesis weights, and other examples of clinical trial optimization that we considered in recent papers include optimal decision rules at interim looks in clinical trials with adaptive designs, as well as optimal decision rules in clinical trials that employ several patient populations. Here is a short list of recent publications that deals with practical approaches to clinical trial optimization and its applications to late-phase clinical trials. This includes a book entitled Clinical Trial Optimization Using R. I have mentioned this book earlier in this course. The book features multiple case studies from clinical trials, and it gives examples of simulation-based calculations aimed at identifying optimal trial designs or optimal analysis strategies, and it relies on the Mediana package. The Mediana package will be the main topic of the next section. Mediana package. We will now take a closer look at this R package. It was first introduced in 2015 with the idea of providing a standardized approach to performing clinical trial simulations. The idea was to create a set of software tools that will help facilitate a systematic simulation-based assessment of trial designs and analysis methods in clinical trials. To accomplish this, the Mediana package was built around the clinical scenario evaluation approach. This package provides a general framework for clinical trial simulations that are typically performed in late phase trials, and this package has been used in multiple clinical trials and featured, as I said, in several recent publications to support clinical trial optimization, which is aimed at identifying optimal designs or optimal analysis strategies. As I said, the Mediana package was released in 2015 and it can be downloaded from the CRAN website. You see the link on the slide. You will also find a detailed online manual, which is very useful if you would like to learn more about this package and its main features, and it's also being used by more experienced users. This online manual was created by my longtime collaborator, Gautier Paul, who has made very important contributions and he has been responsible for several key components within this package. I would like to use this opportunity to thank him and other people who have contributed to this package. It's been a great collaborative effort. If you happen to be interested in contributing to our work on this R package, I would like to encourage you to send me an email. We would love to work with you. Example 3, schizophrenia trial. We will use example 3 based on a phase 3 trial in patients with schizophrenia to illustrate simulation-based power calculations in trials with multivariate multiplicity problems, or in other words, problems with several families of hypotheses. As you remember, since we have looked at this clinical trial multiple times, we are faced here with a bivariate multiplicity problem, since there are two sources of multiplicity. First of all, there are two dose-placebo comparisons in this trial, the low dose denoted by dose L and the high dose denoted by dose H compared versus placebo. Secondly, the efficacy profile of the experimental treatment is evaluated using the following two endpoints. First, the primary endpoint, which is based on the change from baseline in the PANSS total score, and a supportive secondary endpoint, which relies on the change from baseline in CGI-S, a very important index. Our goal here would be to run standard sample size calculations, which means that we would like to compute the number of patients to guarantee a sufficiently high level of an appropriately defined overall success probability. We will talk in a couple of minutes how to appropriately define this performance criterion. The approach we're going to follow in this section is to utilize simulations to compute statistical power for a range of sample sizes and then identify the sample size that leads to a desirable level of power. We're going to run simulations based on the Mediana package. Also, throughout this section, we will follow a general thought process based on clinical scenario evaluation. We will be asking ourselves the following questions. How do we identify the data model and plausible sets of statistical assumptions that will help us define appropriate data model parameters? Or what are the appropriate analysis approaches to be considered in the analysis model, which includes, of course, the most relevant multiplicity adjustment? Or perhaps a question such as, what is the best way to define the overall success probability when we set up the evaluation model for this clinical trial example. The first step in this process is to build a data model. A data model, as you remember, defines the process of generating patient data in a clinical trial. So what we need to do here is to specify a number of parameters of the trial's design. And the general data model includes the following specifications. We see those specifications on the slide. We need to specify the outcome distribution. We need to define samples that are defined as independent sets of patients. For example, samples may correspond to trial arms. We need to specify parameters of individual samples, which includes outcome distribution parameters and sample sizes, and also in certain cases we may need to provide additional information about the trial design. In this particular clinical trial example, 3 samples will need to be defined in the data model, and each sample corresponds to a trial arm, and therefore we'll have samples 1, 2, and 3 that are based on the placebo arm, low dose arm, and high dose arm. The outcome distribution, we will assume that the primary and secondary endpoints are normally distributed and they actually follow a bivariate normal distribution. And as far as the trial design is concerned, we will assume a fixed design. This slide defines the key parameters of the outcome distribution, specifically of a bivariate normal distribution for the 2 endpoints of interest. Within each sample, we need to specify the marginal parameters of the distribution of the primary endpoint, which is endpoint P, and endpoint S, which is the secondary endpoint. Using this information, we can start writing code that will be used to define the data model for this clinical trial example. This slide 22, defines the parameters of the marginal distributions of the primary and secondary endpoints. The information presented here matches the information that we saw a minute ago on slide 21. In addition to the marginal distributions of the primary and secondary endpoints, we also need to provide information about their adjoint distribution. That means that we need to specify the correlation matrix. In this particular case, we assume, based on historical data, that the correlation between the primary endpoint and the secondary endpoint is 0.3. Then we specify an object that gives us the marginal distributions of the 2 endpoints, and once again each marginal distribution is normal. Then all this information is going to be combined to define the 3 objects that are called outcome.placebo, outcome dose L, and outcome dose H. These are the objects that include all of the outcome distribution information. They will be used to define the data model. The data model is defined on this slide, and the first step is to initialize the data model, which means that we need to create an empty model, and then we're going to add the individual components one at a time. In this particular case, after setting an empty data model, we're going to add individual objects that specify the multivariate normal outcome distribution. Then we're going to define the sample size object. This is the object that specifies the common number of patients enrolled in each sample, assuming that we have a balanced design, which means that all samples are assumed to have the same sample size. And we also specify 3 sample objects, one for each sample or each trial arm in this clinical trial example, and each sample object specifies parameters of each sample in this data model. We are done with the data model for example 3, and the next step is to set up the analysis and evaluation models. Within the clinical scenario evaluation framework, an analysis model, as you remember, defines the statistical methods, for example statistical tests that are applied to the trial data generated from the data model. In this case, the key components of the analysis model will include the dose placebo tests and their parameters, and also a multiplicity adjustment. Evaluation models are used in the media package to specify the success criteria for evaluating the performance of the analysis model, so we will define a relevant success criterion. The components of the analysis model are defined on this slide. We will use simple two-sample t-tests here to perform treatment effect comparisons for each endpoint. Multiplicity adjustment will be performed here based on a general gatekeeping procedure defined in Module D that assumes several sequences of hypotheses. You will see the name of the function that implements this general gatekeeping procedure. And as far as the evaluation model is concerned, we're going to compute disjunctive power for the primary and secondary tests, which means the probability that a significant effect will be established at one or more doses on the primary endpoint, and then separately on one or more doses on the secondary endpoint. To specify the multiplicity adjustment, which is one of the components of the analysis model, we're going to use the code that may be very familiar to you. We used exactly the same code when we talked about software implementation of general gatekeeping procedures at the end of Module D. The components of this multiplicity adjustment include the families object, which defines the process of assigning the individual hypotheses to the families. We can see here that the hypotheses H1 and H2 are included in the primary family, and the other two hypotheses H3 and H4 are included in the secondary family. Using the component procedures object, we're going to specify the component procedures. The primary hypothesis will be tested using the Hogberg procedure, and the Hogberg procedure will also be applied to the secondary family. The difference here, of course, is that in the primary family, we're going to utilize a truncated version of the Hogberg procedure, and to specify this, we need to define the gamma object. This object shows us that within the primary family, the truncation parameter of the Hogberg procedure will be set to 0.7. So this will indeed be a truncated Hogberg procedure. And within the secondary family, the truncation parameter is set to 1. So here we will have a full-blown regular Hogberg procedure. The components we have defined on the previous two slides are combined on this slide to set up the analysis model for example 3. We begin with an empty analysis model, and then we're going to add the components. The main component which is added on this slide is the multiplicity adjustment. Here we simply pass the parameters, the objects that we defined on slide 27. The description of the analysis model continues on the slide, and here we specify 4 test objects. Each test object directly corresponds to a dose placebo comparison on either primary or secondary endpoint. We're almost done. This is the specification of the evaluation model. We begin with an empty evaluation model, and then we're going to add 2 components. These are the criteria that are based on disjunctive power for the primary endpoint. Once again, it's the probability that at least one primary hypothesis will be rejected, and it may be meaningful to also take a look at disjunctive power within the secondary family, which is going to be defined as the probability of rejecting at least one secondary hypothesis. This is the last step in the process. The data model, analysis model, and evaluation models have been built, and all we need to do now is to define simulation parameters using a sim parameters object. The object has the following arguments. First of all, n.sims defines the number of simulations. proc.load defines the processor load for parallel computations. Because the Mediana package relies on parallelizing simulations, and if the proc.load parameter is set to full, that means that simulations will be run on all available processor cores, which will definitely speed up the simulations, and then the results will be merged across the cores. And finally, the C defines the seed to be used in the simulations. The 3 models we have built and the set of simulation parameters are passed to the CSE function. CSE here stands, of course, for Clinical Scenario Evaluation. It will normally take a few seconds to run the simulations, and the simulation results will be saved, in this case, to the ex3.results object. To create a simple summary of the simulation results, we can call the summary function, as shown at the bottom of the slide. This table summarizes the results of the simulations based on the mediana code that we have defined. We can see here that with the sample size of 110 patients per arm, disjunctive power, or the probability of success on at least one dose, is equal to roughly 82%. And if we increase the sample size per arm to 140 patients, the probability of success based on the primary endpoint is going to be roughly 90%. In general, with a simulation-based approach to sample size calculations, we rely on a grid search to compute the final sample size in a trial. We typically go over a range of sample sizes, and we identify the sample size that corresponds to the desirable level of the chosen success criterion or probability of success. In this case, this would be disjunctive power based on the primary endpoint. We have run power calculations in example 3 using the Mediana package. As you could see, it's a fairly straightforward process. The code presented in this section is easy to expand to handle power calculations in more complex settings, where we have to deal with several multiplicity components, for example, 3 components based on multiple endpoints, multiple doses, or multiple patient populations. And I would like to quickly add that the Mediana package also provides tools for generating publication quality simulation reports. They are saved as Microsoft Word documents and include a very detailed summary of all assumptions and simulation results. An important feature of simulation reports is that they are easy to customize. This is accomplished by defining the so-called presentation model. With a presentation model, a user can customize the document structure, for example, create sections and subsections within the report, which comes in very handy when we have to deal with very complex data and analysis models. We can specify how the rows will be sorted within tables, and many other options are supported. To find more information about available presentation models and many other features supported by the Mediana package, please visit the package's online manual. Here is once again the link to the online manual. Several useful case studies that illustrate clinical scenario evaluation as well as clinical trial simulations in general can be found on this website. Please follow the link shown on the slide. And case studies include simple trial designs and analysis strategies, as well as more advanced settings that include trials with multiplicity components. I would also like to mention that all Mediana code from our book on clinical trial optimization, including code for simulation-based assessment of clinical trials with multiple objectives, is also available on this website. Thank you so much for your interest in the general topic of multiplicity. We are now done with this online training course. I hope you have enjoyed watching the videos included in this course and working through the examples. If you have any questions, please feel free to contact me. You see my email on the slide. Thank you very much.